Welcome back, everybody, to Chapter 3. As a reminder, this is entitled Cellular Forms and Function. This will be Part 2 of our lecture series for this particular chapter. So if you haven't listened to Part 1 yet, please go ahead and stop, go back, and we'll see you once you have covered those particular topics. Now, for the rest of us, let's go ahead and now that we have become a little bit more familiar with our plasma membrane, and we have discussed passive versus active transport, the fusion versus osmosis, let's go ahead and pick up our discussion and take a look at our next topic, which will cover what we call tonicity. Now, osmolarity and tonicity will work together in this concept. It's based on the fact that our human body is mostly made out of water, so that means that your cells are bathed with H2O molecules, both extracellular and intracellular. And what we see happening is that depending on the environment on the inside versus the outside of the cell, it will regulate how water will move either into and out of the cell. Now, when you discuss the environment that the cell is surrounded by, we like to call that tonicity. And tonicity will take a look at non-permeable solutes, so solutes that are inside and outside of the cell and are not able to pass through the plasma membrane. Now, the solids will not be able to move, but what will be able to move will be the water, the solvent, the liquid portion. And what we want to do is we want to see how the movement of the water is going to affect the overall shape of the cell. So we're going to have three scenarios when we compare the inside and the outside of the cell. And here on your PowerPoint, they're listed, and I want to discuss them with you briefly, and then I'm going to go ahead and give you some examples along the way. So if we're comparing inside to the outside of the cell, and we're taking a look at the amount of solutes that are non-permeable, so keep an eye on that, the solutes cannot move through the plasma membrane. If we're going to compare how many solids you have inside versus outside of the cell, you're going to come across three scenarios. The first scenario is at the bottom of the slide. It's going to be called an isotonic solution. In an isotonic solution, your solutes are going to be equal both inside the cell as they are on the outside of the cell. So you have the same amount of solids on the inside as well as on the outside of the cell. The second scenario is if you have a hypertonic solution. In a hypertonic solution, we see that you have a higher concentration of non-permeable solutes on the outside versus on the inside. So there are more solutes on the outside of the cell than there are on the inside of the cell. And then our third scenario is now going to be called hypotonic. And hypotonic will mean that you have a lower concentration of non-permeable solutes on the outside of the cell than you have on the inside of the cell. So once again, isotonic, everything is the same. Hypertonic, you have more solutes on the outside, so in the environment. Hypotonic, you have less solutes on the outside, more solutes on the inside. Now, keep in mind, these solids cannot pass the plasma membrane. So what we're going to do now is we're going to take a look at how that will affect the water. Will it move into the cell, out of the cell, and how will that then change the cell? Now, as you can see, there's a lot more text on the PowerPoint, but for now, I want to go ahead and do a quick drawing for you. So I'm going to click over to a clean slide over here, and bear with me real quick. I'm going to give you three different scenarios. So I'm going to go ahead and draw three little boxes, and the boxes will represent my outside environment. So this, for instance, could be kind of like a beaker that you're going to fill up with different solutions. And then here in purple, let me do purple, make a little circle. That's going to be my cell. As you can see, I really missed my calling for art school. And then here, using the little X, this is going to represent my solutes. Okay, so once again, you would think I would get better at writing things out, but it's not working for me. Okay, so on the previous slide, I said to you that when we do tonicity, what we're doing is we're comparing what's happening on the inside of the cell to the outside of the cell. So I said to you we have three scenarios. One scenario is when everything's equal. So I'm gonna make 3 X's inside, 3 X's on the outside. So this is my isotonic. Okay? Second scenario was hypertonic. And hypertonic is when I'm going to have more solutes on the outside. So I'm going to draw 6. Then I have on the inside. And my last scenario was hypotonic. And that means that I have less solutes on the outside of the cell than I have on the inside of the cell. So let me draw my little X's. Thank you for bearing with me. Okay, beautiful. So here are my scenarios. Everything is the same. I have more on the outside. I have less on the outside. Now keep in mind, this is all about the solutes, and the solutes cannot pass through the plasma membrane. What will pass through the plasma membrane will be my water, my H2O. And one thing you want to remember here is that when it comes to water, it will always move from high concentration to low concentration. So water, remember, will do osmosis. It's going to go from high to low. Now, you know what's happening with the solute. How do you figure out what's happening with the water? Well, it turns out that the solute concentration is going to be inversely related to your water concentration. And inversely means it's going to be the opposite. So if I say to you, your solute concentration is high, that means that your water concentration is low. If I say to you that your water concentration is high, that means that your solute concentration is low. So they are going to be the opposite of each other. So for instance, I'm going to go ahead and start right here all the way at the outer right-hand side for my hypotonic solution. And as you can see right here, we have a high concentration of solutes on the inside of the cell versus on the outside of the cell. So what I'm going to see is that the water is going to do the opposite. So that means that I'm going to have more water on the outside, making my little circles, bear with me, than I have on the inside of the cell. Okay, so water is high outside, solute is low outside. Water is low inside, solute is high inside. So they're always going to be inversely related. Let's do the hypertonic solution one. Where would you have more water? Would you have more water on the inside of the cell or on the outside of the cell? Remember, don't complicate matters. It's inversely related. So that means that I'm going to have more water on the inside of the cell, because that's where my solute is low, than I have on the outside of the cell. What do you think happens if it's even? Well, if the solute is even, guess what happens with the water? It's even as well. Okay, so now we figured out what the water situation is in each of these 3 scenarios, and now we're going to come back to our first little movement thing that we were discussing, which is that water will always go from high to low. So it will do its osmosis and move from a high environment to a low environment. So that means, starting out again in my hypotonic solution, in a hypotonic solution, I have low solutes on the outside, high water on the outside. I have high solutes on the inside, low water on the inside. So that means that water is going to go from high to low, which means that the water is going to go into the cell because it's moving from high to low. In a hypertonic solution, I'm going to see the opposite. Because I have more water on the inside of the cell, water is going to move out of the cell. And in an isotonic solution, since everything is even, remember, molecules always move, but whatever moves out moves right back in. So I'm going to make a little 0 marking here, indicating that the net influx of water is going to be 0, meaning whatever goes out comes right back in when you take a look at an isotonic solution. Now, what effect will this have on the cell? Well, hopefully you'll agree with me that in a hypotonic solution, when water goes rushing into the cell will swell up, very similar to you blowing up a balloon. And because our cells are just made out of a plasma membrane, you do have the potential that you blow up the cell so much that it literally pops open. It lyses, meaning that it falls apart. If this happens in your red blood cells, we call it hemolysis. Hemo for blood, lysis to pull apart. Your red blood cells will literally pop open. This, for instance, an example of a hypotonic solution is distilled water. Distilled water, which is very commonly used in labs, DI water, is 100% pure water. All right, that means that you have lots and lots of water particles, very low to none solute particles. If your cells come into contact with a hypotonic solution like distilled water, water will have nowhere else to go but from the outside environment into the cell, and this can obviously swell up the cell and can cause some damage, especially if the cell pops open. In a hypertonic solution, you have more water on the inside of the cell than you have on the outside. So water will leave the cell, and when it leaves the cell will start to crenate. Crenate means that it shrivels up. Crenate. A-T-E. This is crenation. Okay? Crenation means that the cell will shrivel up and it will become smaller. A really good example of a hypertonic solution is a salt solution. Our body isn't really made. Let me rephrase this. Our body, when we take a look at our ions, our solutes, the most common one that we have is salt, but it's at a very low percentage. If you were to expose cells to a high concentration of salt, like for instance 5%, 10% salt, what we would see is that the salt will cause the water to exit the cells and the cells will shrivel up. Some people will tell you this is what you see whenever you go swimming, if you've been to the ocean, or maybe if you take a nice relaxing bath. When you look down at your fingertips, sometimes they'll look a little pruny, a little shriveled up. The reason that partially happens is because your epithelium cells, your little skin cells, are in contact with a hypertonic solution. They're in contact with all that salt that's in the ocean, or even if you're lying in your bath, in your bubbling solution that you put in your little bath bombs or whatnot, that has a lot of salt solutes. It drains the water out of the fingertips, so that's why they crenate. They get all crinkly. And last but not least, in an isotonic solution, the shape of the cell does not change. It maintains its natural shape. So whatever goes in comes right back out when it comes to water. There is no net gain in or net gain out. Take a look at these illustrations for our red blood cells. Now, unfortunately, because of a remote situation, I know some of you are taking the 2085 lab also virtually, so there are some virtual activities that most likely you'll do in your lab that will kind of see the movement of water into and out of the cell. But this is very similar of what you would observe if you were doing this face-to-face and you had a little bit of blood sample and you were exposing it to different solutions. If you take your red blood cells and you put it in an isotonic solution, something like physiological saline, the cell would maintain its natural shape because whatever water goes in comes right back out because the concentration of solutes is the same both inside and outside. If you take your red blood cells and you put it in a hypotonic solution, notice how the cell swells up, and that's because water is rushing into the cell. And that's because in a hypotonic solution, there is more water on the outside than there is on the inside. There are less solutes on the outside than there are on the inside. And last but not least, hypertonic. Hypertonic is when water is leaving the cell, and as it leaves the cell, notice how the cell size will be reduced, and it crenates. It gets these little wrinkly edges. And that's because the water is moving from high to low, high on the inside of the cell to low to the outside of the cell. Now, I want to give you an example of why you would kind of care about tonicity, especially for those of us going into the healthcare field. So imagine that you have a patient that comes in, and this patient has maybe, let's go back to the good old days when we were able to socialize and go out. And they had a really nice time over the weekend. They went out a lot. They had some adult alcoholic beverages. They didn't really hydrate enough. So now they're coming into your clinic. They're not feeling well. And it turns out that they're severely dehydrated. Now, many of us will say, okay, that's an easy solution. Let's hook them up to some fluids, right? Let's give them an IV. Let's rehydrate them. Well, when you go to rehydrate them, it's very important to give them an isotonic solution, a solution that is similar to the contents that you have on the inside of your cells naturally. And that's what physiological saline is. Physiological saline is basically a lot of water with about 0.9% sodium chloride, as well as also some electrolytes, very similar to what we are exposed to on the inside of our cells. And that works out perfectly. Now, let's say on this particular instance, whoever was handling the patient made a mistake. And instead of giving them physiological saline, which I'm going to write it down for right here, physiological saline usually has 0.9% sodium, Na. Let's say this person messes up and gives the patient 9% sodium, so about 10 folds more. Well, some of you might go, well, what's the big deal? Well, the big deal is that when you give your patient a 9% salt solution, that solution is a lot saltier than what's happening on the inside of the cell. So when that solution hits the blood vessels, what we see happening is because there are so much solutes on the outside of the cell than there are on the inside of the cell, it's going to allow the water to exit out, and what happens is that the cells will lose even more water because they're going to start crenating, which means that you dehydrate your patient even further than they already are. The opposite would be if you go ahead and give your patient pure distilled water. So what if you give them something that's 0% sodium and 100% water? Well, when you do that, you're giving them a hypotonic solution, my apologies. Hypotonic means that the water goes from high to low and it goes into the cell. And as a result, the cell will swell up and can eventually pop open. So please keep this in mind that whenever you're discussing tonicity, you're comparing the environment on the inside to that of the outside of the cell. And that water will always go from high to low. And the concentration of solute is inversely related to the concentration of water. So when you're doing your homework or you're reading an exam question and it says to you that the concentration on the inside is high, it has a high concentration of solutes, that automatically means it has a low concentration of water. So you don't need to know both. As long as you know one, you know that the other will do the opposite. Alrighty, so let's go ahead and take a look at some other mechanisms of cell transport. This one is going to be called Carrier-Mediated Transport. And this is going to rely on those proteins that you saw mentioned in part 1, where we were discussing how there are different proteins that are embedded within the plasma membrane. And these are going to be very specialized carriers because we can classify them either as a uniport, a symport, or an antiport. And these are going to be almost like little pathways, little tunnels, and they are going to be able to regulate which solutes can move in and out of the cell. Now, I've inserted a little picture for you right over here. So for those of us who are more visually inclined, we can see the difference between the different carriers. So on your left, it says that if you're looking at a uniport, una means one. That means that you're moving one type of solute. So you can see the little arrow is moving just one little particle, and this can be both inside or outside of the cell. I mean, towards the inside or towards the outside of the cell, depending, of course, what you're moving. So in this example, it tells you, for instance, if you're looking at a pump that is specific for calcium, if it's only moving calcium at any time, it's called a uniport. Now, what if the pump or the carrier is able to move multiple solutes? Well, we're going to call that a symport. A symport can move two or more, but also it has to move in the same direction. So here on my illustration, I can see in green, I can see I have two different solutes, and notice how they are moving together. And it's important to point out that anytime you see the symport, that they have to move in the same direction. If you're going to have them move at opposite direction, then we're going to talk about an antiport. And an antiport, as you can see right here, this is our yellow. You can see that you have, once again, at least 2 or more solutes. And notice how one is being pumped in while the other is being pumped outside. So uniport, you just move one solute. Doesn't matter if you're moving in or out, but you're only moving one specific particle. Symport is you're moving 2 or more, but you're moving in the same direction. So we're either all going into the cell or we're all coming out of the cell. And antiport means 2 or more solutes, but you're going in opposite direction. Probably the best example of an antiport carrier-mediated transport is your sodium-potassium pump. Your sodium-potassium pump is essential for our homeostasis because it allows us to regulate our sodium and potassium levels. It tends to kick sodium molecules out and pumps potassium molecules back into the cell. Your body spends a lot of ATP maintaining the sodium-potassium pump, so it is an example of active transport. There are also some other examples that I want to discuss with you when it comes to cell membrane. This one is going to rely on a little carrier, a little vesicle, instead of a little protein to move it from point A to point B. So let's go ahead and let's take a look at that one next. All right, so this is going to be called vesicular transport, and like I said, it's going to rely on a little vesicle. And a vesicle, think of it like a little bubble container, kind of like a little Ziploc bag. So whatever we're transporting, we're going to kind of build ourselves a little membrane around it, so it makes it easier to move it into or out of the cell. Now, if you see that the vessel is moving into the cell, we call that endocytosis. Endo means to enter. If it's moving out of the cell, we call it exocytosis. Exo, obviously, for exiting. Now, as you can see on your diagram, on your slide, you can see that under endocytosis, it says we are going to use a vesicle to bring materials into the cell, but we're going to have different options. We can do phagocytosis, penocytosis, and receptor-mediated endocytosis. So what's the difference? Well, it all depends on what you're transporting into the cell. Phagocytosis, phago means hunger. This is going to be called cell eating. This is whenever you have a large entity, like a solid particle, that's being engulfed into the cell. Penocytosis, peno means drinking. This is whenever you have fluids or droplets or minute little particles that are entering into the cell. So phago is for the large solid entities. Peno is for the liquid items that are moving in. And then receptor-mediated endocytosis means that you have a particular receptor that the particles will have to bind to before they can actually enter the cell. Now don't worry, I do have some examples for you coming up so we can take a look at these particular processes and get a better view of it. Let's start with this one right here. So here, this is a very old school illustration, but what we're looking at is a little macrophage, a little white blood cell, that's this little blob. These little circles, these are little red blood cells, and as you can see, the little blob is chasing a small little piece of black vibrating entity, you see? That's a little bacteria. And what's going to do is it's going to chase it, and eventually it's going to catch it. And when it catches it, notice how it literally eats it. It surrounds it. There you go. It swallowed it. That, ladies and gents, is phagocytosis, which is an example of endocytosis because it's entering, and it's phagocytosis because it's a solid entity that it's allowing to swallow and enter into the cell. And in fact, this is happening in your body right now. We have white blood cells that scavenge our bloodstream and also go into our tissues and just look for anything that does not belong. And one of the ways they alert our immune system is that they'll do phagocytosis, they'll bulk eat it, and thereby destroy it. Here's an illustration that comes right out of the book. So we can see right here, we have our cell, and notice how they went ahead and they told you it's a phagocytotic cell. Not all cells are able to do this, but a phagocytotic cell will be able to do phagocytosis, bulk eating. And in this example, you can see it has a little particle that it wants to interact with, so it will form a pseudopod. Remember that from part 1? When it will interact with the environment, it will extend its plasma membrane. It will surround the particle and it will eat it, engulf it. And notice how it puts it around a little vesicle, a little container. So that's what makes it endocytosis, and more particular, because it's a solid, it makes it phagocytosis. Once it's inside the cell will use things like lysosomes and digestive enzymes to destroy the particle, whether it's doing it for nutritional values or just to kind of get rid of it. If it's something that can harm the body, it all depends on what type of cell we're looking at. And then look what's happening over here. So you can see step number 4, it fuses with the lysosome. Step number 5, you have enzymes that will digest, so they'll destroy the little particle. And then look what we're doing in step 6 and 7. Once the particle is destroyed, you don't want to hold on to the waste products. So you're going to notice that the vesicle, the little container, makes its way over to the plasma membrane and then exits the cell. Well, when it does that, it's doing exocytosis. So endocytosis is when it's entering, and when it's entering, remember, it's using a vesicle, which means that if it's entering with food or solids, then it's phagocytosis. With liquids, then it's penocytosis. And when it exits, we call it exocytosis. Here's an example of the receptor-mediated endocytosis. So that means that in order for the particles to come in, they're going to have to interact with receptors that you can find on your plasma membrane. It's also only when they find the receptors that the vesicle will then form. Notice how it pinches in, so it's going to use the plasma membrane to make a little vesicle. And that is when the particles can enter inside the cell. So they rely on the receptor before they can do endocytosis. A fun fact, the HIV virus, the one that causes HIV and AIDS, it's actually a receptor-mediated model, meaning that when it tricks your T cells, your white blood cells, to engulf it so it can get access to your DNA and replicate itself, it actually has to match up to certain receptors before it can do endocytosis. So currently, one of the ways that scientists are combating the disease is to come up with a vaccine that will hide these receptors from the HIV virus. So if you hide the receptors, that means that the HIV particles will never be able to find the receptors. And if they can't find the receptors, they can't be engulfed into your white blood cells. And because that means that they won't have contact with the host cells, that means that the virus will no longer be of any harm to us, because a virus needs a host cell to survive. So just a little FYI why these receptors can be so important. Alrighty, so now we come to our last topic of chapter 3, and that's going to take a look at the organelles. So the organelles are mini organs, and these are going to be specialized structures that we find inside of our eukaryotic cells that are going to allow us to have specific functions for each. And this is in essence what makes our cells so complex, but yet still energy efficient. So we're going to take a look at just a handful of them, not all of them, but we're going to take a look at the most popular ones, including the nucleus, the mitochondria, the Golgi complex, and the ER or endoplasmic reticulum. Here's a quick reminder. You saw this picture on part 1 of what your typical cell looks like, and we discussed the fact that it has a plasma membrane, that's the border. So now what we're going to do is we're going to concentrate on the inside and see what all these little organelles have to contribute to the functioning of the cell. And I think it makes sense to start with probably the most important one, which is going to be right here in purple, and that's going to be our nucleus. All right, so here's a look at the nucleus. As you can see, it has a nuclear envelope that allows it to be separate from the rest of the organelles within the cytoplasm. Now, for the nucleus, go ahead and remind yourself that this is where we house our DNA. So this is where you have all your chromosomes and your genetic information. DNA cannot leave the nucleus. So that is why we're going to see that the nucleus will have lots of nuclear pores, little openings, because DNA will transcribe itself into RNA, and RNA can freely pass through the nuclear pores and go in and out of the nucleus as it pleases. But DNA, your genetic code, stays inside of the nucleus at all times. Inside the nucleus, you will also have the nucleolus. And the nucleolus is essential because that is where you're going to be producing your ribosomes. Your ribosomes are essential for protein production. So the nucleus houses the DNA, and then your nucleolus, which is inside of the nucleus, is where you produce or synthesize your ribosomes. Here's another look at the nucleus. So as you can see, they've marked for you your chromatin, which is your coiled up DNA. You can see your little nucleolus floating around in the center. You have your little nuclear pores that your RNA can use to go in and out of, and that's because DNA has to stay in, but RNA comes right back out. Now, the reason we started with the nucleus is because since that houses all our genetic information, that is the first location where every cell has to go to for any type of instructions that it requires. So for instance, let's say your body is interested in producing a particular hormone, let's say estrogen or testosterone. Its first step will be to go ahead and cause the cell to go within the nucleus and activate the gene, the particular piece of DNA that's required to produce the protein that you're after or the hormone that you're after. Once you have your directions in DNA, you will transcribe it into RNA. RNA will leave the nucleus and will then interact with our next organelle, which is going to be the ER, our endoplasmic reticulum. And as you can see, it's very close in contact with the nucleus. Now your endoplasmic reticulum, your ER, there are two of them. You have your rough ER and you have your smooth ER. And as you can see from the picture, the rough ER is the one that's going to be studded, covered all over by these little purple dots, which are our ribosomes. The smooth ER does not have any, hence the name. Now, the covering of the ribosomes will tell you exactly what the rough ER does, and that's because one of its main functions will be the production of proteins. So the rough ER will help you with the production of any proteins that you might require. The smooth ER, its primarily function will be for lipid synthesis, so it will help you make any fats, any lipids that are required. It also produces detoxifying enzymes, so it's a great way that your body can utilize it to get rid of any type of pathogen or any type of toxins that are around. It will rely on the smooth ER to produce some of those detoxifying enzymes. So the cell started off in the nucleus. It got its directions from the DNA due to the nucleus. Then it went over to the rough ER and it picked up any proteins it might need. And then it went over to the smooth ER. It picked up any lipids or fats that it might require. And now what we're going to do is we're going to put those proteins and fats all together so that we can produce our final product. And that's going to involve another organelle. For that one, we're going to have to go over to the Golgi complex. Some of you might know it as the Golgi apparatus. Both names are correct. The Golgi complex, as you can see right here, it's a series of flattened sacs. I like to informally call it the UPS store of the cell, and the reason I say that is because all your individual ingredients, like your proteins, your lipids, will come together, excuse me, and be mixed into their final format within the Golgi apparatus. So this is when you get your final product. So the individual ingredients go in, and the final products come out through the little vesicles. You see the little containers right here, and these can be filled with things like the particular hormone you needed, a particular enzyme, any type of secretion that the cell relied on. And once that's produced, of course, then now you can decide if you want to keep that particular product inside the cell and use it there, or you want to do exocytosis and allow it to exit. So for instance, a lot of times the hormones will do exocytosis and they'll be dumped into the bloodstream to travel over to the target cells. If you have, for instance, a lactating organism that you're looking at, the milk being produced will be dumped into the milk ducts and it goes over to its area of interest. You also have some housekeeping organelles that we definitely have to mention, two of those being the lysosomes and the peroxisomes. These will just be floating around in the cytoplasm. Lysosomes get their name because they're able to lyse, which means they're able to pull apart any type of entity you no longer need. So it's essential for things like malfunctioning organelles, or maybe just old organelles, any type of waste, any type of pathogen that might have been introduced into the system, into the cell cytoplasm. We can use lysosomes to lyse them. You particularly tend to see lysosomes in areas or cells that have a lot of filtration rates. So for instance, like your kidney or your liver, they will be particularly multiple there because they're great housekeeping organelles. Another thing that you can rely on are peroxisomes. Peroxisomes get their name because they are able to generate hydrogen peroxide. So you can see them right here floating in the cytoplasm. And what happens is whenever they come across certain toxins, primarily blood toxins, they're able to generate hydrogen peroxide, and that is then one way that your body can rid itself of a toxin and maintain its homeostasis. And then the last one to mention, I think most of you have heard of this one before, the good old mitochondria. There you go. So the mitochondria is the powerhouse of the cell. This one is FPL. And the reason for that is because the mitochondria is where we're able to convert our energy that we generate, for instance, from our food, into chemical energy, aka ATP. So you saw ATP before because we discussed the fact that your body relies on that for active transport. So the ATP comes courtesy of the mitochondria. Now, a few interesting things about the mitochondria, and you can see that on the illustration. One is that the mitochondria actually has two membranes. It has an outer membrane on the outside that's nice and smooth, and then it has an inner membrane that is right below the outer membrane that's full of these little curves. And the reason it has two membranes is because once upon a time the mitochondria was a free living entity, meaning that it was a cell all by itself, and it was engulfed by a larger cell which became its host cell. I like to think of it kind of like a little Pac-Man. So you had a little cell that was eaten by a larger cell, and the little cell, instead of it being completely pulled apart, became an important organelle of the larger cell. Now, before you start thinking, that's crazy, that can't happen, remember there's some proof behind this. The first proof is the fact that it has two membranes. The inner membrane belonged to the original cell, and the outer membrane was placed there by the host cell to now declare it as belonging to itself. The other piece of evidence that we have that the mitochondria was a free living entity millions of years ago is the fact that mitochondria actually has its own DNA. It has mitochondrial DNA. Mitochondrial DNA is unique because you can only find it in the mitochondria. None of the other organelles besides the nucleus have DNA. They simply rely on directions from the nucleus to get their work done. But mitochondria can fully function by itself. However, it does collaborate with the nucleus to make it effective for the body. And here's the kicker. Mitochondrial DNA comes exclusively from your mother. So the DNA that's in your mitochondria right now is identical to the DNA that your mother has. The DNA that's in your nucleus is both from mom and dad. So both parents contribute to nuclear DNA. So just a little FYI. So the mitochondria is our powerhouse. It produces ATP. It's part of the endosymbiotic theory, which basically said that once upon a time it was a free living entity. And our evidence for that is the fact that it has a double membrane and its own DNA. And for those of us who've heard of the endosymbiotic theory before, we might recall that there's another organelle out there that's also part of it. We don't have it, but plants and any organism that photosynthesizes does, and that's good old-fashioned chloroplast. Chloroplast is another organelle that has been proven as part of the endosymbiosis theory. It has its own DNA, and it has a double membrane. All right, so having said that, I think this is a nice place for us to end chapter 3. As always, please take your time, go over the notes, and if you have any questions, comments, and concerns, please do not hesitate to reach out, email me, and please interact with your classmates by utilizing the posted discussion threads. All right, we will talk soon. Our next chapter will be chapter number 5, where we're going to take a look at histology, the study of tissues. Until then.